Jag är inte så säker på att jag tittar på det virus.
Då är det nästan bättre att titta på svamp.
Det finns ett väldigt intressant anekdot där. Hon tittar på hur virus och piggyback på bakterier polioviruset. Då är det så att polioviruset bara finns där. Ganska svårt att komma in istället. Det finns ju efter dem kommer på ett sätt som de inte vet. Så åker de med när bakterierna lokaliserar sig.
Ja, men vi borde ju...
Just nu så håller vi på med just när vi tittar på det här arkeologiska Nej, inte helt i alla fall. Det finns ett antal bibliotek för att
Kommer jag paddan där. Köra trådlöst.
So, welcome to Friday's afternoon research seminars, and today I have the pleasure to invite my collaborator Ignacio Rangel from Örebro University, and he works in this Nutrition Gut-Brain Interactions Research Center. And the title of this presentation would be Pathways of Interaction of Microbe and Gut-Brain Interactions. So, Ignacio, welcome.
So, hello. Okay. Okay, so thank you very much indeed, everybody. I'm really happy to be here in Kalmar. Thanks really a lot for the invitation. Thank you, Ravi, for inviting me to the department, to the colleagues that I have already met. It's really a pleasure, as I said in the morning to the students. It's the first time for me here in Kalmar, and it's such a warm welcome, not because of the weather, but because of the discussions I've had so far with you all. So I'm really keen in having some discussions with you all and presenting a little bit my part of what we are doing, what I'm doing in my research, and I mean a little bit the wider perspective of this so hot topic, which is at the moment this microbe gut brain axis. So I work at the Örebro University, at the School of Health and Medical Sciences, and my research is under the research center, which is called Nutrition Gut-Brain Interactions. Now I know that I have to use this one. And a little bit about first the center. So NGBI, as we call it, is a research center that was established in 2012, and we got funds from the Knowledge Foundation and the industry to establish this research center. And the idea, or the main goals, as you can see there, is to get an overview of this interaction, the nutrition, microbe, gut-brain interactions. And of particular interest at that time, and still, is the association to intestinal disorders. And our models, let's say, of disease are IBS, irritable bowel syndrome, IBD, inflammatory bowel diseases, and we are very interested in what's happening with aging. We are expanding our interest in this interaction into the more psychological, neurophysiological disorders. And there we're interested in different aspects, such as depression, autism, ADHD, even Parkinson's diseases. And as I said, a key part of the work at the moment is having a kind of second shape or push is aging, health and aging. I mean, we are getting older. How can we cope with a healthy aging process? So you're very welcome to visit our homepage, and afterwards to visit us physically. You are always welcome in Örebro. We don't have that much snow, unfortunately, but you are always welcome to be there. Fine, and this is what we used to present, how we think about our interactions. And we think about the process from nutrition, microbes, gut, and brain. And today I'm going to focus on the part which is related to my own research interest. I'm a macrobiologist, and my main focus has been in the last 8-10 years in the microbial ecology of the gut. So we're interested in this part, the microbes and the nutrition, because we're interested in the influence of the fibers in the production and the composition of short-chain fatty acids in the intestines. We are interested in looking at fibers and short-chain fatty acids specifically on the metabolism of the microbiota. Then we're also interested in looking at the interactions, microbe interactions, and then some of the most important points that we are looking at the moment is the gut-brain function, the gut barrier function, leaky guts, which are typical of IBS and IBD diseases. And of course, I mean, the immune function. What is the effect on the immune system? And going from the microbes to the gut, what do we have in the gut? What the microbes are doing, and how they interact both with the host and within them. So these are, widely speaking, some of the questions that we tend to pose when speaking about the microbes in this interaction. And why are we so interested in the microbes? Why I'm so interested in microbes? And I used to say that I'm interested in the microbes because whenever I'm in front of an audience, as many of my colleagues said, what I see is microbes sitting, listening to me. And that is because we know that basically we have more than 1,000 species in the gut, and I'm meaning different microbial species. Phylotypes, we're talking about roughly 1,500 different phylotypes. The numbers are not increasing that much, but they are still, I mean, depending on the depth of the analysis, can be growing, I mean, to some extent, but not that much. But what is extremely interesting is that we have 10 more times bacterial cells in our bodies than our eukaryotic cells. That's why I say that, I mean, I see microbes sitting in front of me. The same applies also for the genes and the genome. And I think we tend to think very little about that. It's not only the microbes per se, but there's 100 times more or fold more genes that we have in the microbial package than the eukaryotic ones. Then we can imagine the capacity, the genomic capacity that we are carrying with the microbial composition. And they are, of course, fundamentals for different processes, and that's something very well known. I mean, fermentation for the immune system. They are the ones producing certain vitamins, and they are helping us against some pathogens by different ways. What I mean with this immune system training is one of the, I think, most exciting and relevant parts, I mean, when speaking about the microbiome. This is a so-called germ-free isolator. It's from a colleague, Christoph Reinhardt, that when he was at Sahlgrenska in Gothenburg, was working with these kind of systems. So with these kind of systems, I guess you are aware of it, what you can actually have is animals that are so-called germ-free. That means that they are completely devoid of bacteria or organisms. So you can have them and raise them in sterile conditions, which is unique. There are no organisms, at least on this planet, that are completely sterile. And what is really interesting with these systems was, I mean, what could be tested and shown on these animals. So these germ-free animals are very special. So they have no bacteria in the gut. And when looking at the gut vascularization of the intestine, the small intestine, what Christoph and Fredrik Beckhead found was something like this. So on this side, you have the villi, intestinal villi from germ-free mice. On your right side, you have the villi from conventionally raised mice. That means those that have been grown, the same kind of mice from the same mother, but not from the same mother, from the same batch, but grown conventionally, in non-sterile situation. And what you see is a huge difference in vascularization that can be measured here on Biopic software, just for measuring the width of the vascularization. And you see here the differences between the germ-free mice and the conventionally raised mice. So germ-free mice have a disturbed vascular system in the intestine. And what is even more exciting is that these guys, what they did was, okay, so now let's take the microbiota from these guys and place them on these ones and see what happens. And this is what happened. So they checked the vascularization on the conventionally raised mice, on the conventionalized mice. That means with bacteria placed into these germ-free mice for 14 days, and this is what they saw, a recovery in the vascularization. amazing, isn't it? That's what you can see here. So the differences, and this is the vascular density per villus. So that told us quite a lot about the importance of the bacteria in training the system, even the physiological system. And that's why, I mean, there is, for example, a lot of interest now in microbiota and cardiovascular diseases, thrombosis, because there seems to be links between people with coronary inflammation and a particular microbiota composition, and even some metabolites associated to that. If we look at now, for example, I said the importance of the microbiota. Now, for example, for the immune system, if we look at how it develops the microbiota composition throughout the years or throughout, I mean, in infancy, this is something that we did with children, with some colleagues in Gothenburg, looking at children from 1 week of age up to 12 months. And these are just specific groups of bacterium that we were looking. Just this is for E. coli. And the interest was to see, are there any differences in these children if they are born by cesarean section or vaginally? And this is what we found. This is for vaginally born babies. This is one for cesarean section. And you can see the difference. There is a difficult trend to explain here for the vaginal ones, but vaginally born babies. But what you can see is the bacteria present, E. coli, a very typical one. Cesarean sections babies, I mean, they have a very jumpy pattern of bacteria for E. coli. Something similar happens also with Bacteroides. So this is the vaginal born babies. These are the Bacteroides one. Sorry, the cesarean ones for Bacteroides, and you see the same trend. Lower trends. I mean, they're recovering step by step, like here, from week 1 to 12 months. So really, even for example, the birth way is affecting also the microbiota. For some of the bacteria. For others, surprisingly, it's not. For example, bifidobacteria. We got, I mean, colonization-wise, similar percentages in the babies, either by cesarean section or vaginally born, except for a point here, which is difficult to explain, but it's more or less even. So when we looked at the total microbial composition, now it's not single bacteria, but the total microbial composition. This is an example of after 6 months what we saw. These points are for vaginally born babies. These are for cesarean section babies. This is PLS, partial least square method, to multivariate method to split out groups on a supervised system, supervised method. What you can see after 6 months that these babies are still, most of them are grouping apart from the vaginal ones. What happens after 12 months is that finally you can start seeing a kind of mixture. So the babies born after by cesarean section it takes long time, 6-8 months for them to recover and come back to the situation where we have the babies vaginally born. It's a fantastic development. So what we can say is that then, of course, the delivery mode is pivotal for the development of the microbiota. That after 12 months, babies are kind of recovering, depending on the cesarean ones. And that what is interesting is that it doesn't apply to all the bacteria. It applies to some bacteria. Our example is based on E. coli and Bacteroides, but not other bacteria. The question is why? Why these bacteria, why not others? And what is their role in it? Right, so I move on with other cases. I mean, this relevance of the bacteria or the microbes in association to the gut. This is for the a study in health. What happens with the gut microbiota in health and disease, the standard situations? We have as as a standard example for our system this IBS disease, irritable bowel syndrome. Irritable bowel syndrome is a disease that is on the rise. And Western countries are really struggling with that. So this is a picture showing the complexity of the disease. It's of course a disease resulting in inflammation in the intestine. It's not that chronic and that hard as IBD, as Crohn's diseases or ulcerative colitis, but it is a result of actually of a combination of processes between the host and the microbes. I don't plan to go through the details here, but what I want to say is that what we know is that there seems to be a potential effect of the gut microbiota. Changes in the gut microbiota are affecting the physiology in the gut. And what we see are changes, as I'm going to show in some minutes, changes in some of the immune cells. But what is also very interesting is that this is a complex disease which is associated also to, for example, genetics. We always tend to look at genes. We want, historically, that has been the way to go, isn't it? Obesity, which genes are associated to obesity? Cancer, which genes are associated to cancer? We can continue with disease after disease after disease. And it's been really difficult, actually, in my opinion, see what you think, to have an association of this particular gene or SNP to this particular disease. But still, there are some studies indicating soundly that genomes or the genes are associated to IBS. But stress and diet are also very important in the development of IBS. This part is, of course, one of the key horses, the driving horses in our research, stress, how stressful reactions affect the gut functioning, not only the brain functioning, but also the gut functioning. And we know that antibiotics and stress in the form of early child experience are also associated to IBS in the future. Similar to, for example, these kids that I showed you, the children with born by cesarean section or vaginally, some of them are more prone to develop autoimmune disorders, the ones born by cesarean section. So within this pattern, the interest is to know, are the microbial composition in healthy individuals different to the ones in IBS? And the answer is yeah. So we run a study where we check the microbial composition in IBS individuals, here marked in red, compared to healthy individuals. And there is some overlapping. But there are some individuals really varying from the standard kind of microbial composition from healthy individuals, as you can see here. When looking at something which is very important for determining the status of the microbiota diversity levels, what we found was the following. We collected samples both from the feces or fecal samples, and also something that is very important, the mucosal. So we had biopsies from those individuals. So these first three set of points are from healthy individuals, individuals with IBS from the mucosa, the biopsies, and these ones from the feces. And we had two groups of IBS patients, so-called general IBS, and people that develop IBS after an infection, after a pathogenic infection. Very likely, diarrhea. They have problems with food poisoning due to a pathogen. And what you can see here, in both cases, for the mucosal and the fecal composition, is a decrease in the diversity. And that's one of our landmarkings. I mean, diversity is good. So high diversity, the composition is stable or is more homogeneous. Low diversity, that means that something has happened in the system which is making some organisms to survive better or to increase in numbers. And it was present both for the mucosal and the fecal samples. When looking then at what was happening between the microbes and the immune cells, then we looked at this lamina propria lymphocyte. That was on the samples on biopsies. And you can see that the diversity levels were, this is in post-infectious IBS individuals, that the diversity levels were negatively correlated to LPS generally, and also to naive T cells, these CD8 plus cells. There was a negative correlation with these kind of cells. So again, talking about the importance of, in this case, the diversity and the immune responses. So we think again back on this axis is the gut, or the microbes in the gut, and in the gut we specifically see what is happening with the immune system. I showed you this part with the vascularization, which is not necessarily about the immune response, it's physiology, but I can tell you, I mean, that in these kind of mice, one sees similar trends. I mean, the immune system is completely not destroyed, but it's very unbalanced. And now in individuals, we see a similar trend. So different changes in the microbial composition and in the diversity, changes in the immune system or the immune cells. The question that we think is more important is: are there differences? Yes, but or not. But what are those differences? Where are those differences? And then we were searching in the of hunting. What are the good guys? What are the bad guys? So, and by applying also advanced statistical approaches, we targeted samples from healthy individuals, again, our post-infectious IBS individuals and IBS patients. And what you can see here is this clustering of individuals based on their bacterial composition, microbial composition. And on the one side, individuals with post-infectious IBS were pulled out. Their microbiota was dominated by these Bacteroidetes proteobacteria groups. While on the other hand, on the other side, the more IBS healthy individuals were more dominated by Clostridia. Particularly two groups of Clostridia, Clostridium clusters 4 and Clostridium cluster 14A. These are specific groups and important groups because a lot of what we call butyrate producers, short-chain fatty acid producers, are placed in these groups. And that was a nice hypothesis because higher number of these bugs from these groups are associated to healthy individuals, but also to IBS. That was a bit of a tricky point. But what we decided to do afterwards is, okay, if we take those bacteria and then we use those bacteria to stimulate the host in the form of biopsies and an ex vivo system, what will happen with the response of the immune system in the form of inflammation? So what we did was to treat biopsies, colonic biopsies, and stimulate them with gut bacteria. And then we looked at the effect on the inflammation on the cytokine level. This is in short a summary of what we found. We use different bacteria, Ruminococcus navus, Ceobacterium luminosum, Subtuligranolum variabile, and we found changes in some cytokines, pro-inflammatory and anti-inflammatory cytokines. And I don't have time to go in detail through that, but some of the cytokines, I guess the ones that you know are more aware of are these, for example, IL-1 beta, which is a pro-inflammatory, strong pro-inflammatory cytokine. And what we could see from these guys is that they increase actually the levels of IL-1 beta in the tissues. We looked also with samples from post-infectious IBS, again with other bacteria. Interestingly wise, I mean, one of our favorite bacterium, Akkermansia mucinifila, and there were some changes also in some of the cytokines, particularly this IL-13. Again, IL-1 beta was also showing differences between the unstimulated and stimulated biopsies. It's a lot of information here, but the moral of the data is that with particular bacteria, with a specific bacteria, one can modulate, change the immune responses in an ex vivo system. Whether that is transferable to a higher level is more difficult to say. So, and now moving to this with the microbiome in diseases, with different diseases. So nowadays it's known that looking at the microbiome, in particular different diseases, obesity, IBS, colon cancer, and so on, if we take samples from all those different patients, group of patients, the microbiota is going to be different. That is what is shown here. And in recent years, it was also shown, it has been also shown that it is also applying to diseases such as autism, schizophrenia, anxiety. Anxiety is very interesting because, for example, as I showed you in the picture of IBS, a lot of patients with IBS are more prone of suffering anxiety disorders, depression. So going through different processes that a non-IBS person is going, probably the anxiety levels are more manageable. With an IBS person is very sensitive. So triggering the IBS is a result, in many cases, of depression. So here, this is a superb system to work on the gut-brain interaction. Unfortunately for the patients, luckily for the researchers. But we have been interested in that part. And you have seen tons of examples of what is happening, I mean, now between these two systems, the gut and the brain. And there are lots of public, populistic mentions about it. But, I mean, I just took, for fun, I mean, a couple of examples. One is from this Forbes magazine, saying the gut feeling and how intestinal bacteria could manipulate your brain. And it says next time you feel like craving for something like chocolate, it may well be your microbiota and not your cell that are actually craving for that. And it's a populistic approach, but that was based on this paper. Is eating behavior manipulated by gastrointestinal microbiota, evolutionary pressures and potential mechanisms? So there are some patterns really tested indicating that how the microbiota may be tuning brain functioning. An example similar to this one, which I always love presenting, is this one in the New Yorker that said, the E. coli made me do it. And that is based on a story in Canada where there was it was raining like madness, like snowing in Kalmar the 2nd of March. And what happened is that in a small town in Canada, they suffer floodings. So the pipelines, they just started, they couldn't cope with all the water, in the draining water, and they was starting coming out. So people in the town, in that particular town, started suffering of infections. And some very smart scientists afterwards kept an eye on what was happening with the population. And so they went on to ask the people after this problem with the rain. And they were asking a question, after this outbreak, when they were having infections, had you been told for the first time by a doctor that you have developed depression, anxiety disorder, panic disorder, or post-traumatic stress? And most of the answers were yes. So from an infection disorder, people afterwards showed problems with anxiety and depression. And again, that was based also on a scientific paper. It was picked up by the New Yorker. And I think this is based on you have seen many of these pictures, I guess. But this is based on this complex communication between the brain and the intestine. And the brain is pumping down a lot of information to, for example, the gut via some accesses, this high HPA access in the form of hormones such as cortisol. And these hormones are actually targeting some particular cells, but also interact with the bacteria in the gut. And the bacteria in the gut are reacting back by also communicating with the immune cells, as I briefly showed you in the beginning of the presentation, and also by sending, for example, signals such as the way of tryptophan, and the way in the form of short-chain fatty acids. And what we know now is that they are also producing all precursors for neurotransmitters. And I will just mention a bit about that in a second. So one of our favorite compounds to look at is within these short-chain fatty acids is butyrate, which is a short-chain fatty acid, small molecule, very low molecular weight. But the effect of it seems to be quite interesting. So what we have been doing is we have been looking at these butyrate-producing bacteria, because they are an excellent model to look in the microbial composition in the gut. But looking particularly at butyrate, this is the bunch of bacteria that can produce butyrate. What I was saying in the morning to the students is think like, I mean, the identity of these bacteria varies quite a lot. So evolutionarily, what has happened is that the capacity to produce butyrate has been transferred to bacteria from different lineages, which is also, as a microbiologist, quite intriguing. From our favorites, some of our favorite bacteria, again, I mean, are represented here like Faecalibacterium prausnitzii. But butyrate is produced by in different ways or different pathways. These are the most important ones. The most relevant one is this acetyl coenzyme A. But it's very. It has been shown that it can be produced by these other patterns, and there are bacteria that may not have this pattern, but they have others. Anyway, so coming back to the point of butyrate, why it is important? It has been shown in vivo by using or applying enemas daily to individuals, 100 millimolar enemas to individuals for 14 days, that it has targeted some antioxidants or antioxidative processes in the form of this glutathione. So it has affected the antioxidative processes in situ. We have known about that in vitro, but not in situ. So what we know is that these butyrate producers phylogenetically are very dispersed, that they can be produced by different metabolic pathways, and that they act at the oxidative stress levels in situ, which is also something that was not known until the outcome of this paper in 2010, I think, it's already. So our interest in these short-chain fatty acids and butyrate has been on can it have an effect at other levels? For example, at the cell level, what are the mechanisms behind this behind these short-chain fatty acids? So in collaboration with Ravi here from the department, your department, what we have been doing is using a cell model. Fibroblasts from healthy individuals have been stimulated with butyrate and in the presence of an antioxidant, and looking at tryptophan uptake, and using this hydrogen peroxide as an oxidative stressor. And what Ravi and company have been seeing is the following: that measuring the tryptophan uptake on this axis by looking at different concentration of butyrate with the presence of this stressor, that with only the stressor, there is a decrease in the transport of tryptophan. And there seems to be a kind of recovery process of butyrate by coming back to levels of the controls at the different concentration of butyrate. So there seems to be then a protective effect of butyrate on the transport system of fibroblasts in healthy individuals. So if that is the case, it has to pass through the intestinal barrier. That's what we think. So what we have been doing is to search for butyrate receptors and transporters at the gene level in intestinal cells. So we have been using a cell line as well, CACO2 cells, colonic epithelial cells, and treating them in the same way with butyrate, and looking then for transporters and receptors of short-chain fatty acids. And what we have been seeing is that, indeed, again, this is hydrogen peroxide and then hydrogen peroxide in different levels, that there is, we can find these receptors and transporters in the colonic cells. So the receptors and transporters for butyrate are present as well in the colonic cells. So we want, of course, to look at these receptors and transporters now in the fibroblasts, and something which is even more exciting, which is glial cells. Glial cells are representative of the brain functioning and also representative of the ENS, the enteric nerve system, because that will cope very nicely with how a particular microbial metabolite such as butyrate can interact with the second brain, which is the enteric nerve system. Fine, and what we are trying to do is actually making a model, a whole model, where we look for mechanistic approaches in the way of looking, for example, for fibroblasts, glial cells, ex vivo systems by using biopsies, and also finally in vitro, in vivo systems. So, and we are always recruiting people. So you are welcome to contact me if somebody is interested in lying on a bed and receiving some butyrate or something like that. It's quite fun, actually. You receive lunch and breakfast, and you can watch TV. So quickly, to round it up, probiotic bacteria, something that we have been doing with probiotics. And this is on IBS. This is a cartoon of more or less how it should look like. I mean, in the intestine. I mean, the barrier function. This is the colonocytes, the lumen, and the mucus, and all the digesta and the bacteria. So one idea is, well, what happens if we try to change the microbiota by adding one new bacterium? And what happens if we actually add more bacteria, different bacteria? So we have been adding different bacteria, lots of lactobacilli, propionobacterium, and bifidobacterium. And again, I don't have time to go through all the results, but what we see is that actually the probiotics, this is again a cluster analysis. What you can see here is what's happening with the healthy individuals receiving the probiotics, and then IBS people before and after receiving the probiotics. And probiotics are not changing dramatically, actually. If we only look on this group of people, they're not changing dramatically the microbiota. So that is also something very intriguing. So you add new bacteria to the system, at least in the doses that we have been using, and they don't change dramatically the composition of the microbial composition in the microbiota. So we know that substantially they don't separate them, they don't change or modify it. But the question is, what does this cocktail is actually doing at the immune level and also at the brain functioning level? And that is something that we are doing at the moment in the research center, looking at this, trying to answer these questions. So part of this stuff that is within this approach of from the brain to the gut, which is extremely complex, we speak about the microbiota, and I've spoken a lot about the bacteria. And I was telling in the morning the students that there are two groups that we have overseen throughout the years. And now is not the case, but I mean, there are two groups that in my opinion are extremely interesting. One of them is fungi, and the other one are virus. Fungi are becoming more important than just Candida albicans or skin fungi. They are becoming very important from the point of view also of immune responses. And one which is extremely exciting is the virome. So all the viral particles that are actually helping the bacteria, for example, in reproduction, are becoming very interesting, for example, in IBD. So there are some viral particles that have been associated to inflammatory processes, particularly IBD. I want to finish up with this part. So when we speak about the microbiota, this is the presentation, the cartoons, oversimplified cartoon of the intestine. And we used to think about this, the bacteria that are in the lumen, in this luminal composition. The point is, are we really looking at the right bacteria? So the bacteria can be here in the luminal part, but there are some bacteria that can be also in the mucosal part, the ones that can attach actually to the mucous layer. And those are the ones that actually can be more important, more relevant in particular cases. And what we did was to look at the composition of the microbiota in healthy individuals and in IBS, both in the mucosal level, biopsies, and the luminal content in the form of fecal samples. And what we can see here is that for the case of the healthy individuals, the microbiotas from both sides more or less overlap. There are some individuals that really have different bacterial compositions in the lumen and in the mucosal. But in the case of disease, such as IBS, this is what we found. The microbial compositions are very different. Very, very different. And this is what we also could see here on this heat map analysis of the bacteria, particularly for example, Clostridium clusters. So what you can, basically you don't need to check this, but I mean the colors for the biopsies and feces, how it's completely twisted. So the higher numbers that we see in the feces disappear or are very, very low in the biopsies. So everybody that is interested in the microbiota of a particular disease or in healthy individuals, I think we have to ask us the question, what are we looking for? Is it the microbiota that is associated to the lumen, or the one that is associated to the mucosal? And why is there any difference between them? And is it important to ask it yes or no? And the answer to me is yes, because if we are interested, for example, in metabolic processes affected by the microbiota, then probably we are fine with the with the luminal microbiota. But if the important is, for example, the immune responses, then we are, or the brain interactions, then we are probably better off by looking at the mucosal microbiota. The majority of the studies, having said that, are based on fecal microbiotal compositions. So that's maybe a question to pose to everybody, and next time we see what is happening there. Fine. So I think I can just summarize this presentation by saying that briefly about what I told you, that the microbiota is a dynamic part of the body. So we showed it with children, and what I really believe, and I have been discussing with some of our colleagues here, is that if you come to me, you are very welcome to leave samples every week. And I bet that your microbial composition will change from 1 month to another month and another month. And it would be even more fun if you can leave blood samples, because then we can look, for example, at changes in some hormones. And so I'm not meaning that the microbiota changes from 0 to 1. in a scale from 0 to 1. I mean that within a range, within an inter-individual range, there are variations. And we need to understand those variations to be able really to cope with for example, understanding some diseases that are very, very variable, such as, for example, IBS. IBS is one of the most difficult diseases from the point of view that people are very sensitive. And some measurements shoot up to the roof in 1 week, and the second week they are just down close to the floor. Butyrate, which is a microbial metabolite, helps against oxidative stress, and we have shown it in mechanistic studies with fibroblasts. Hopefully, we can go now to further analysis with also with, for example, glial cells to get closer to neural cells. The probiotics, what about that? Well, they have a limited impact on the composition of the gut microbiota, but we know that they have an effect on the immune response. So what we're doing now, and I didn't have time to present this, we're looking at probiotics with different number of pili. The same bacterium with lots of pili, same bacterium with a standard number of pili, a naked bacterium. And what we can see is that there are differences in the response depending on the pili numbers, not the composition, the pili numbers. So we're really coming down to the point of even protein level and what are the mechanisms behind probiotic effectivity. Right. So I have lots of collaborators, but these are few of them. One of them is Ravi, working here. Another one that started actually the research center and it has been the leading force in the NGBI is Robert Brummer. Well, I have collaborators also in Sahlgrenska and Wageningen and some collaborators also in Finland, also working with the microbial compositions. Fine. So that could be a short summary, very, very short summary of part of what we do in Örebro. And you are very welcome to visit us anytime. Thank you very much.
Thank you, Robbie, for the nice introduction, and thank you, Ignacio, for a very, very nice talk here. So this paper is open for comments or discussions. Do you have any ideas? I can start.
Yeah.
So a research friend of mine, Julie Pfeiffer in the US, she's working with germ-free mice, and she has shown that poliovirus more easily can get into the mouse model system if they piggyback on E. coli. And one thing that you only briefly mentioned is the importance of fungus and virus. It would be nice to see, because there is also so many viruses that are there everywhere, but nobody knows why they are there. And whether you can see that when you do your NGS sequence library measurements, or if you just throw them away as garbage.
I think that's an excellent point, and I think that has been the mistake. I mean, the depth of the sequencing has been stopping most of the time at the level of sequences that can cover 16S. So and they are very well targeted, but the unknown sequences, I mean, if you go for deeper than 2 million sequences, or above that, then you lose a lot of the viral particles. And that has been, I think, the problem. As I said, there are, at least that I am aware, 2 or 3 groups now deeply looking into the virome in association with, as I said, IBD, particularly IBD. And the papers they have published are very sound and extremely interesting. But it's a little bit, I guess, the problem that we have with bacteria at the beginning. There are lots of viral particles that I mean, the identity levels are high, but how do we understand them? And I think I'm not a virologist, but I mean, it's even more difficult. I know that you are into virus. I mean, how do you work with the virus, I mean, to really understand what they are doing? We have this problem with bacteria and also with fungi. So with the viral part, I mean, it's a real challenge. But having said that, as you commented, I mean, there are lots of viral particles that are found in the genomic sequences in bacteria. But until now, I mean, people haven't really been able to understand why they are there. If it's not just a transport system.
Okay. Comments or not? Ideas? Hopefully we will hear from you more because your ongoing collaboration with Ravi here. So
yeah.
Please come back and tell us more about this because it's a very interesting topic.
Thank you.
Let's give Ignacio another warm hand. And for those of you that are members of the department in chemistry and biomedical sciences, there will be a seminar at 2:30 by the newly recruited professor in environmental science, Professor Marcelo Ketzer. Next Friday, there will be no seminar. The seminar is postponed. The Friday after, we have a dissertation, Emma Ahlstrand. But the Friday seminar after that, the 23rd of March, we have Professor Björn Lindecke from Uppsala Universitet and Apotekarsocieteten. And he's going to talk about the chemical analysis of the medical coffins they found on the wreck Kronan outside Öland. That will be a very interesting topic. So have a nice weekend and see you within some weeks then. Bye-bye.